Hello, everyone, and welcome to today's webinar, Spatially Defining the Future of Cancer Biology with Advanced Multiplexed Immunofluorescence. I am Megan Pascal of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Ultiview. To learn more, visit ultiview.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have trouble seeing or hearing the presentation. I'd now like to welcome our speaker, Troy Bloom, Senior Field Application Scientist at Ultiview. Troy, you may now begin your presentation.
Thank you very much. Thank you, everybody, for joining today. Very nice to be here and giving you an overview of the Ultiview technology. So let me share my screen. Bear with me 1 minute. And let me go into presenter mode. Okay, here we go. So just a little bit about myself. Again, my name is Troy Bloom. I'm a Senior Field Applications Scientist at Ultiview. I have over 30 years of experience in the field of histology, especially in IHC and ISH assay development. I joined Ultiview almost 2 years ago in November of 2020. And then prior to joining Ultiview, I was the manager of the histopathology core lab at a company called Acceleron Pharma. So let's go over today's agenda of what we're going to cover today. So first thing is, how a high throughput, high-plex, multiplex immunofluorescent assay can more accurately predict treatment response. From there, we will go into whole slide image analysis of the tissue and can provide insight into the tumor microenvironment. Then we'll move into how image analysis can be applied to discover cell types, populations, and morphological context. And then finally, we'll do an application case study, okay? So moving forward, in this slide right here, everybody has seen a diagram of tumor microenvironment complexity. As we all know, the tumor microenvironment is the environment around a tumor. This is also going to include the surrounding blood vessels, immune cells, fibroblasts, signaling molecules, and the extracellular matrix. The tumor and the surrounding microenvironments are closely related and will interact constantly. Tumors can influence the microenvironment by releasing extracellular signals. They are promoting tumor angiogenesis and inducing peripheral immune tolerance, while the immune cells in the environment can affect the growth and the evolution of cancer cells, okay? So, with the complexity of the tumor microenvironment and it's driving the need for multiplex markers, of course, as we all know, everything is going to start with a basic hematoxylin and eosin stain. This is where morphology is going to determine diagnosis. And as informative as an H&E stain is, of course, it doesn't really tell us exactly what those cells are expressing. So, from there, the gold standard is simple DAB-IHC, right? And from there, usually we're going to do single protein detection. And then from there, we can start to do cell identity. Or start to do diagnostic algorithms to predict biomarkers. And as great as the DAB stain is, of course, at most you can do 1-2 markers at a time. But, there's a lot more information in these tissue samples that we're working with. So how do we get there? So now we go from singleplex DAB stain into a multiplex immunofluorescent stain. And then from there, obviously we can start looking at multiple protein detection. Then from there, get the deeper cell phenotyping with the higher expression range, and then, of course, intracellular interactions and the networks around them. So with an overview of tissue-based multiplexed methods, with that, the efficient translational research, therefore, is again going to depend on concentrating resources and molecules and methods more likely to succeed as biomarkers. While balancing risk of discarding potential valuable candidate devices, prototypes too early in the development process. So mid-sized panels appear to be the best methods to keep you in what sweet spot where you want to be able to generate big enough data sets with reliable data to help identify the best biomarker candidates, right? So as you can see, with number of markers. You could start off with CyTOF maybe or DSP, and then start deplexing down to a UltraView technology or TSA-based technology, and then, of course, move it right into a CDX environment. So just an overview of the UltraView technology. It's a very simple process, right? So after sectioning your slides and baking your slides, you move right into the full protocol. First off, you're going to start off with a single antigen retrieval step. With the UltraView technology, we use a EDTA TRIS-based buffer, pH 9.0. The protocol is carried out for 20 minutes at 100 degrees C if you're using a Ligabond RX. After antigen retrieval, you then move into the application of your antibodies. All your antibodies are going to go into a single cocktail, applied all at the same time. So from there, after you've applied your antibodies, we then go into our amplification step. This, of course, is a single step. This is where we amplify the signal of each of the unique DNA barcodes that are attached to each one of the antibodies in your cocktail. And then from there, we go into our detection step. Attached to each one of these probes is a unique fluorophore. The fluors that we use are spectrally distant from each other so that you don't have to utilize any spectral unmixing software after the fact to deconvolute the signal and get your ability to visualize the signals within each and every cell that you're staining, okay? So moving forward, talking about some of the available options that we provide to our customers. We have three offerings at this point in time. The first one is our fixed view kits. We have 7 altogether. From there, you can then move into a more flexible kit, which is what we call our FlexView kits. This is based off of our 8-plex technology and our fixed view panels, where we allow our customers to swap out up to 2 markers and select up to 2 markers from a list of 12 markers that you see on the right-hand side. And then, of course, if you want to do a truly custom kit, we have those capabilities as well. You can select up to 8 markers, whether from some verified markers that we currently have in our portfolio, or if there's a marker of your interest that we don't currently have, we can definitely apply that into whatever plex assay that you would like to do. But what we're going to focus in on today is our 8-plex fixed view kit, which is what we call our Immuno 8 panel, okay? So with this panel, of course, you can stain up to with 8 markers. The 8 markers that are in this kit are CD3, CD4, CD8, and CD68, along with FOXP3, PD1, PDL1, and pancytokeratin SOX10. On below that, you can see that you can start to put together a multitude of phenotypes depending on what your interest lies. Here you can see there are about 14 phenotypes that we're showing here. And again, regardless of the tissue type or the disease type that you want to be able to apply these panels to, our technology is very agnostic. So as you can see here in this slide, we're showing non-small cell lung cancer, ductal carcinoma in situ tissue, and of course, colorectal tissue. One of the nice things I like to point out about this slide here is that all of the tissues that you see here were stained with the same kit during the same run. So of course you can see the flexibility of the kits and how it can be applied to multiple tissue types. And again, even though our kits are mainly focused in the immuno-oncology field, you can utilize our kits in any disease indication that you would like to apply it to. So now looking a little bit deeper into the predicate comparison of the staining with the markers here, the next couple of slides will cover over all the markers within the Immuno 8 kit. So at the top row, you can see we're focusing on 4 of the markers in the 8-plex kit, which is CD8, PD1, PDL1, and CD68. You can see here the DAB staining, and then from there we move into our single-plex ISP. ISP stands for our technology, which is in situ-plex. You can see the correlation between the DAB and then the single-plex staining, and then we then take an image from our multiplex staining as well, and then you can see the correlation between the 3 staining runs that we did there, of course, across all 4 of these markers here. And then moving forward on the next slide, we look at the other markers in there as well. And then, of course, the same thing follows. Start off with DAB, follow it up with a monoplex ISP, and then move forward with this same image in the multiplex image, just to show you that the staining is consistent for each one of the comparison that we did here. Now, when we start to show some additional data, when we start to look at intraday assay reproducibility, in this slide, we're going to start looking at single marker positive cell densities. You can see that, with most low variables can be observed for cell counts within one run. In this instance, we had some slightly higher values for the CD68 markers here. But you can see that everything is very nicely put together here in nice capabilities. Now, with our single marker positive cell intensities, of course, with that, we're going to be able to measure the positive cell intensities present within lower and more even variable across the markers, suggesting that, variability observed in cell counts is, in part, explained by the biological variabilities or the limitations from the chosen image analysis, okay? Now, moving forward to look more into some of the phenotype subsets that you can utilize using our immuno technology. On this slide, you can definitely start to explore the dynamic nature of the tumor microenvironment. So on this slide right here, you can start to look at the different subsets that you can utilize with our kit, whether it's going to be Tregs, exhausted T cells, cytotoxic T cells, or any of the other ones that you see on there. But moving forward, once we start to look at some of the subsets, so on this slide, we're going to look at some PN T cells, which are going to be shown in CD3. But then if we want to look at T helper cells, we can then look at CD3 and CD4 positive cells. From there, we can start looking at the Tregs and then add FOXP3 into there. And then, of course, if you want to look at your tumor marker, which is going to be the pan-cytokeratin marker, you can observe all of those markers within the tumor microenvironment and be able to start moving forward with your analysis. Now, if we start looking into some of the other phenotype that you can look at, again, we can look at the pan T cells with CD3 positives, move into the cytotoxic T cells where we add in the CD8s. From there, we can start looking at those exhausted T cells where we add in CD3 with PD1. And then, of course, start looking at all four of the markers together with the cytokeratin marker to be able to, again, look at those populations within the tumor microenvironment. And then, if we want to start looking at some real checkpoint markers, we can start looking at the immunosuppressive macrophages where CD68 and PD-L1 exist. From there, we can then start looking at the immune-evading tumor cells with cytokeratin and PD-L1 positive. And then, of course, again, if you want to look at all three of those markers together within the tumor microenvironment to give you a nicer picture of what you're really seeing before you move into analysis. So with this technology, it's been very popular with a lot of our customers and researchers. And recently, we had a paper that was posted this year in 2022. This was posted in the Journal of Immunotherapy and Cancer. This was a paper that was put out by the University of Brussels. And basically, what they're doing here is that they utilize our immuno technology to start doing analysis on 5 advanced melanoma patients using both pre and post-treatment core needle biopsies. Again, these samples were stained with our fixed view immuno kit. And then what they found out is that following treatment, the increase in cytotoxic T cells densities and regular T cells, but then they also saw a decrease in the exhausted T cells and immunosuppressive macrophages. And then with individuals with complete remission, an increase in T lymphocyte infiltration was observed. So all this information was then from repetitive biopsies. And then, of course, the biomarker for intratumoral treatment strategies with the signatures of the adaptive immune response on early tumor samples were highly predictive of the response to treatment. So this was a very nice paper that came out, and it showed the capabilities of our technology when applied to true tissue samples from patients going through therapies. So how do we leverage multiplex immunofluorescence? Well, this is where we start to enter the spatial biology. So, we're going to start answering 3 questions. The what, which is going to be the spatial resolved expression of RNAs and proteins with varying degrees of plexing and retention of tissue cytoarchitecture. Then the Y, which is going to enable the interrogation of complex phenotypes, functional states, and intracellular relationships with help of computational biology and pathology methods. And then we go into the how, which is going to borrow concepts and statistical modeling techniques from geography and ecology, right? So now at Ultiview, we are a spatial biology company, definitely specialized in spatial phenomics. We're definitely positioning ourselves around leading this push into the spatial phenomics area. With that, it's definitely going to provide discipline of mining tissues, images in their own context to identify spatial patterns that are related to clinical outcomes and involves every step from start to finish. So this is going to include development of the assays, analysis of the images, and then, of course, providing the spatial biology, going further into data mining, and then, of course, validation of those findings, okay? So, again, with Ultiview, we are able to support your journey through the entire developmental pipeline, starting at the beginning with defining and framing your specific scientific question and developing your panel of markers, all the way through to delivering answers that enable your project to move forward. Again, we can begin by helping you define your specific question. And then once we understand your specific question, we can begin developing your assay, whether you want to choose from the available panels or see how one of our FlexView or custom assays may be able to answer your specific question. Now, each one of our kits, whether it's a fixed view, flex view, or our UView catalogs, are pre-optimized, and they are to develop to produce quick turnaround times, producing optimized and clear signals for your images. We can do this with up to 8 biomarkers on whole slide imaging with clear cellular resolution. And again, these high-plex stains show you detailed phenotypic profiles and the localization of these cells within their microenvironments. And our assays are gentle enough to run a true H&E stain on the same slide, so you can have that classic pathologist annotation, as well as the detailed high-plex analysis that provides you with a large amount of cellular information. So, we can take all of this information and use images or data analysis to look more closely at the biology happenings within these tissues. Now, as we discussed before, we can do up to 8 biomarkers, but in this example here, we're going to show you a piece of tissue that we've stained up with a 12-plex assay that we currently run in our services lab. Again, we're going to be looking at 12 biomarkers on whole slide images, along with the H&E stains with pathologist annotations. Again, this is going to provide us with a lot of data points for analysis. Now, we can start with the classic H&E for pathologist annotations to determine tumor regions. However, with accompanied multiplexed fluorescent images, we can dig a little deeper, right? So we start with a simple DAPI showing all the cells within the region, and then add the tumor regions in red. This, of course, is a run on the whole slide. So again, whole slide is shown in the upper left-hand corner. And we have the overlay showing the analyzed cellular localizations in the cell phenotypic profile in the circle, which is in the lower left-hand corner, okay? Now we want to start looking at T cells, which are, again, going to be shown here in green. And they're going to show you that some have infiltrated into the tumor regions, which you can see within the white circle. And some have remained in the non-tumor compartment, okay? So if you look outside of the tumor compartment, you can see that you're going to see some of those T cells expressing out there. And with any additional markers, we can start to differentiate these T cell populations a little further. So now we bring in our cytotoxic T cells, which are going to be shown here in light blue. And this is paired now with the macrophage stain. And now the macrophage stain, which is now here in purple, which can show these immune cell clusters, which have been primed by antigen-presenting cells and are within the tumor regions in position to attack those tumor cells. But what if a PD-L1 stain was applied, which is now shown here in yellow, and now we can see these tumor cells have responded by expressing PD-L1, which is then going to be categorized as within the immune tumor cells, and then these patients might be a good candidate for an anti-PD-L1 therapies, right? So with these stains, along with the proper analysis, you can give a much better picture of what is occurring within the tumor microenvironment. So as this captures here, you can definitely see utilizing image analysis. You can start to segment these cells, be able to categorize them, and then again, move forward. And then, as we can do this on the real cells, we can actually see or quantify all of these cellular interactions within the tissue. Here's just highlighted 5 markers to answer one particular question. As I mentioned before, these tissues was actually run with the 12 biomarkers, which provides you with even more data and a clearer picture about the tumor microenvironment and exactly what is happening within the tissue. So you can have a better understanding for drug discovery or patient treatment, for example. Now, again, here are just some of the examples of what we can do with all of this data. Just run a simple whole slide cell quantification, which can then provide you with whatever amount of information you want to provide, whether it's going to be total cells, how many of those are tumor cells, which ones are the T cells, how many of those are cytotoxic, which ones are macrophage, and then, of course, if you're interested in PD-L1, how many of those cells are expressing PD-L1. From there, we can then generate a detailed phenotypic analysis for all of the cell types present. And then we can break the tissue up into compartments. In this example here, we'll start breaking it up into tumor versus non-tumor, okay? And then using these defined tumor and non-tumor regions, we can start to look at the cell populations within those regions. This is going to highlight the importance of location data. For example, information about the immune cells infiltrating the tumor borders can help determine whether a tumor is classified as hot or cold. And this is going to indicate whether it's a potential for targeted therapies. However, we don't just have to stick to just tumor versus non-tumor. We can then start to segment it into more detailed regions and analyze the cell populations within regions like this example here. We can split the stroma up into 3 different immune evading populations, a cold or non-infiltrated region, which is going to be shown here in the blue. And then, of course, after that, we can start and either import structures, for example, shown here in red, or cell clusters shown here in the green, okay? Now, from there, we can start to look at cell clustering throughout these tissues. Here, there are 2 different tissue regions with similar cell numbers. However, in the region on the right, where T cells can spread out and generate more cell-to-cell interactions and a larger cell response as opposed to a more clustered T cell population shown on the left. Now, the spatial data is key to determining exactly what is occurring in the tissue. So distance analysis can provide insights into how active the immune response is in a tissue. In this example, a regulatory T cell shown in the gray can regulate T cells shown in green, and as long as they are in the same vicinity. The T cells which are outside of the region or not close might be activated and uninhibited and free to roam and kill tumor cells. So all of these analysis can tell you a lot about your tissue. From there, we are then able to summarize with an expert analysis and a report and help get you specific answers. In our image analysis packages, we provide quite a bit of information for you to be able to utilize to make the determinations on your research. Starting with same slide fluorescent and H&E image fusion. From there, we can then do whole slide image analysis, which includes multiple marker phenotyping. If you were to do a 4-plex, we can provide up to 8 phenotypes. If you were to move forward with an 8-plex, you can definitely do up to 16 phenotypes. From there, we can also provide you with tissue segmentation, which will definitely be like tumor versus non-tumor, utilizing our CK-SOX10 cocktail antibody. From there, we can definitely provide a spatial phenomics report with the standard readouts, including cell quantification, cell densities, marker intensities, and for whole slide tissue or tumor versus non-tumor. So with the spatial phenomics, visualization of complex phenotypes using anatomic pathology workflows, again, quantitation and spatial analysis of cell phenotypes. Then we can do correlation of cell phenotypes, densities, and proximities with tissue morphology. Follow that up with discovery of new cellular and tissue biomarkers, and then, of course, training of artificial intelligence, deep learning networks to improve interpretation of H&E slides. And that is pretty much the end of the presentation. So I will hand it back over and we can start answering any questions.
Great. Thank you so much, Troy, for your informative presentation. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, does UltiView provide recommendations for the best fluorophore combinations for co-localized markers that are expressed within the same cell? For example, CD3 and CD8?
Yeah, great question. So we can definitely provide you with the right fluorophores to utilize. Again, with the UltiView technology, because we use the 4 distinct fluors that are spectrally different from each other, they're going to help you visualize those cells more easily and clearly. And then from there, of course, we can definitely work with you to work with the best combinations of either fluors or pseudo colors moving forward. But again, the fluors that we currently use are FITC, TRITC, Cy5, and Cy7, along with DAPI as the nuclear markers.
Great, thank you. My next question, have you tested other sample types beyond FFPE, for example, fresh frozen?
Yeah, great question. Currently at UltiView, our assay is developed mainly around formalin-fixed paraffin-embedded tissues. We have not currently worked out a working process for frozen tissues. However, we have worked with various customers who have utilized our technology on their frozen samples, and they have provided us with great input and have shown some positive data that has come from some of that staining. So,
awesome. Is there a better way to stack my sequential images beyond using DAPI?
Yeah, so currently at UltiView, we have a software that we've developed in-house, which is what we call our UltiStacker software. And what basically happens here is that you basically take each round of your stains, and the software will stack these images utilizing the DAPI signal or the nuclear content signal from each one of the rounds that you've stained for. So in an instance of an 8-plex, it'll take the first round of staining and then the second round of staining and then line up the nuclear stain on both of those rounds to get that composite moving. And then, of course, if you wanted to come back and do a true H&E, you can then stack the third round with the H&E together with your first 2 rounds of multiplex immunofluorescence staining, and then, of course, get that nice composite of the multiplex with the H&E.
Great. It looks like we have one more question here. If I have my own IHC qualified antibodies, can UltiView develop a custom kit?
Yes, we can. As I said earlier, with our truly custom UView offering, you can choose either one of our verified markers from the panel, or if you have one of your own IHC qualified antibodies, we can definitely implement that into whatever plex assay that you would like to have developed.
Great, and it looks like that brings us to the end of our Q&A portion of the webinar. Thank you so much, Troy. Do you have any final comments for the audience?
All I'd like to say today is thank you for your time. We really appreciate it. Hopefully, this was very informative for you. And then, of course, if you have any questions, please reach out to us. We are readily available to answer any questions and engage you in any conversations with any future research that you would like to utilize the UltiView technology on.
Great. Thank you again, Troy, for your time today and your important research. We would also like to thank LabRoots and our sponsor, UltiView, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.